Good morning. Before we will begin, I would like to put on a disclaimer that since we are in an online setting, we won't be using the actual reagents utilized in a formal laboratory setting. However, even though this is the case, we will still be executing the procedures with safety precautions, hence the usage of complete PPE. To begin, I would like to formally start with the documents needed in the laboratory. First would be request forms. Specimens are accessioned by giving them a serial number along with request form. Before that, let us first define what request forms are all about. Request forms must include the following. Request forms are one of the most important documents in the laboratory and is gathered along with the specimen. Request forms indicates a patient's data wherein it includes patient's name, patient's age, patient's sex, and date of birth. Take note that if a patient is housed, you must include the patient's room and the bed number in the request form. Lab accession must be plastered along with the request forms, including the specimen type and specimen source. Example, if the specimen is acquired from the finger, you must include which hand and which finger it was acquired. Second of the most important documents acquired in the laboratory is the patient's report. Take note that the patient's report includes the same elements from the request forms with an addition of the diagnosis of both the gross and microscopic findings. After which, attach the image of the gotten sample from both the gross and microscopic findings, including then the name of the pathologist. Patient's report may be forwarded in the form of the following. Moving on to the third document acquired in the laboratory would be incidents report. These kind of reports would guide the laboratory staff in other future problems that they would soon encounter. In addition, request forms are kept for as long as 5 to 10 years or even longer due to the improvements of specific databases. Moving on to the receiving portion of the laboratory. This is where we receive the samples from the patient, and this is where we check the labels as well as the request form. Take note that specimen accessioning is performed in the receiving area. It is important that we check both the label and the serial numbers have matched. Request forms are checked properly due to the fact that this is where most of the errors in the laboratory would begin. Serial numbers are provided in this stage. This portion indicates AP for autopsy pathology and SP for surgical pathology. The last two digits are meant to indicate the digits of the year. This part indicates the specimen control number from the specimen. I will now proceed with the different techniques of smearing.
Smearing is the method of choice for examining sediments and is used for psychological examination such as in diagnosing cancer. So first, I will be showing you the materials that I will be using today. This is my specimen of choice, which is a pig's blood, my applicator stick, and my microscopic slide. So the first technique that I will be demonstrating is the streaking method. Streaking is a procedure in which a selected specimen is applied gently and rapidly throughout the slide in a zigzag manner in order to obtain a relatively uniform distribution. So first, I will pour a small amount of the specimen, the pig's blood on one end of a clean glass slide. Then, in a zigzag manner, I will streak the specimen up until the whole end of the slide. And this is now the streaking method. Now, I will proceed with the technique, or I will demonstrate the spreading technique. It is a procedure in which a portion of a specimen is spread gently into a moderately thick film by teasing the strands apart using an applicator stick. It is recommended for smear preparations of bronchial aspirates, fresh sputum, and thick mucoid secretions. The materials that I will be using is my specimen, an applicator stick, and a microscopic slide. So first, I will be transferring a small amount of specimen on one end of the clean glass slide. Next, I will be spreading the specimen in a straight line up until on the remaining length of the microscopic slide. So this is now spreading. So now I will be performing the squashing method of fresh tissue preparation. Squashing preparation can be performed simply by crushing a small piece of tissue between two microscopic slides. And it is important to know that the tissue specimen must be less than a millimeter in diameter. So first, I will be placing my tissue specimen on the microscopic slide, and then I will cover it with another microscopic slide. Then, I will crush the tissue specimen so that it will be flat. So this is now a squash preparation of tissue specimen. So now I will be demonstrating the pull apart method. So it is a procedure in which the specimen is dispersed evenly across the surfaces of two slides, and the slides will be pulled apart with a single uninterrupted motion. It is used for bone marrow aspirates. So here are my materials, the specimen of choice, which is the pig's blood, into microscopic slides. So first, I will transfer one drop of a specimen on one end of a clean glass slide. And then I will cover it with another clean glass slide in order to allow the specimen to be spread evenly across the surfaces of the two glass slides. then right after covering it with another clean glass slide, I will then spread evenly across, or then with a single uninterrupted motion, I will pull the slides apart. So this is now the pull apart method. So the last technique that I will be demonstrating is a touch or impression smear, or commonly known as imprint. In this procedure, I will simply touch the tissue onto the clean glass slide. And the most commonly used specimen is core bone. However, for this activity, I will be using an animal liver. So first, I will pick up my specimen. Then I will allow it to come in contact with a clean glass slide. This will disallow the cells to come in contact directly with the slide. So this is now imprint.
For the first step, we will describe the specimen. The specimen is displayed fresh in a container labeled with a patient's name, chicken, medical record number of 1, 2, 3, 4, 5, and designated as liver. It consists of a single soft fragment of red plant tissue. Measuring a 4 cm from superior to inferior, a 0.5 cm from anterior to posterior, and 8.5 cm from medial to lateral. The capsule is smooth, blistering, and unremarkable. Serial sectioning from superior to inferior reveals no mastication or prominent nodules. The specimen will be subsectioned and entirely submitted to processing. For the next step, we're going to put the specimen into certain size that will fit into the cassette size. Since you'll be using light microscope, the specimen size should be around 2 cm, 2 square centimeter, where its thickness should not exceed 4 mm. The outer dimension should consider the size of the cassette of 3, 2.5, and the height of 0.4. We will proceed to the next. For the next step, we are now ready to put our tissue specimen into the tissue cassette, ready for the fixation process. Here we have our tissue specimen with the patient's name chicken with the medical record number 12345 designated as liver, our tissue cassette, forceps, and the pencil for manually labeling our tissue cassette. First, we have to prepare our tissue cassettes by labeling them with the patient's name chicken and the record number 12345. Labeling the specimens accurately is important so that interpretation of results will become reliable, at the same time to avoid wrong diagnosis and incorrect treatments. Now that we have labeled our tissue cassettes, we are now going to put our specimen inside the tissue cassettes, sealing them ready for the next step.
So for the next steps of fixation, we will be needing a small piece of foil, a glass with distilled water which will serve as our fixative, and our tissue cassette. And now, I will be submerging my tissue cassette in the fixative, which is distilled water. Make sure that the cassette is fully immersed in the fixative. Next, I will then be covering the glass with a piece of foil and then leave it for 18 to 24 hours.
After we are done with our fixation process, the next step of our tissue processing is the dehydration, because simply the dehydration will remove the excess water from the tissue prior to the wax impregnated. So the first thing to do is we are going to transfer our sample to a 70% ethanol for 30 minutes. And then we will cover it. Next is we will transfer our sample for a higher concentrated solution, which is our 80% ethanol for 30 minutes. And then we will cover it. And then the next step is we are going to transfer our sample again to a more higher concentrated solution, which is our 95% ethanol for 1 hour. Then cover it.
Since we submerge the tissue in an ethanol during the dehydration phase, it is necessary to remove the alcohol from the tissue through a process referred to as clearing. So, the alcohol is removed with a fluid that is miscible with the paraffin wax and with which the tissue must be impregnated. So, the procedure is known as clearing because the tissues are rendered translucent in addition to being free of alcohol, since the refractive index of the clearing agent is nearly equivalent to that of tissue proteins. So, how can we know if the dehydration is complete or not? So, by pouring saline on the tissue section will determine whether or not the dehydration is complete. So, the saline becomes milky if the dehydration is not complete. Thus, the tissue sample should then be placed in ethanol during in an ethanol solution again. So, if the saline does not become milky, we must move on to the clearing process. So, the most generally utilized clearing agent is saline. Nevertheless, we should be cautious when handling the tissue specimens that have been submerged in the saline for an extended period of time, because long immersion in saline might cause tissue distortion. So, our first step is to put our tissue specimen or our cassette into the first saline solution for about 20 minutes. After which, we will transfer our cassette to the second saline solution again for about 20 minutes. And then finally, after 20 minutes, we will transfer our cassette to the third saline solution for about 45 minutes. So after 45 minutes, we will get our cassette and we will put into the paper towel to dry. And after the clearing, the tissue should look transparent.